Welcome to the Biomedical Research Institute in New Jersey, a nonprofit organization dedicating to advancing medical research while educating new generations of students about the importance of science.
So why are they running? Shouldn't they be in the lab?
Good question. Through this fundraiser, the past interns of BRInj, as well as current staff, are helping to promote the importance of research, as well as to give more young people the opportunity to engage in cutting-edge research. But I won't steal all the glory. Let's let the interns tell you all about our new lab. My name is Greg, and I'll be your tour guide through this wonderful new research facility we have. We'll start here. In this room, we have our microscopes that are state-of-the-art, and we use these to visualize things we can't see with the naked eye. In fact, we have one of our interns here, Emma.
Hey, Greg.
Emma, what are you using for this tour today?
Well, I'm looking at cells from a brain primary cell culture that I did last week.
That's interesting. And what's on the screen here?
Well, here you can see two types of cells. You can see neurons in green with their nuclei in blue, and you can also see astrocytes in red.
Well, what are astrocytes?
Astrocytes are very important cells because they support neuronal activity. And I'm studying the interaction between astrocytes to better understand neurodegenerative diseases like Alzheimer's.
Well, that's great, Emma. Good job.
Thank you.
Now we'll move on to our translational research laboratory. This is the largest of our labs here, and as you can see, our research scientists have their offices here, which allows quick access from switching between reading primary literature to aiding their mentees without having to hover. Here we have one of our research scientists, Austin, who's going to tell us what he's doing today.
Doing an ELISA.
What's an ELISA?
It's an enzyme-linked immunosorbate assay.
And what exactly do you use it for?
We use it to detect levels of proteins in cells. Here is an example of what one would like.
Well, that's really interesting.
Yes.
And that's your data there?
This is the data that will appear when I put this into the machine. It'll read the levels of color from the detecting of the protein levels.
And here we have our biomedical research laboratory. Here's where we culture and handle all our primary neuronal cell lines that we use for our experiments. We keep them in incubators like these that offer optimal conditions for growth. And when we handle them, we do so in a sterile environment like the hood to my right. Dan, why don't you tell everyone what you're using today?
I'm using a cell culture model of neuroblastoma to study neurotransmitters.
And what kind of applications does that have?
It has applications for movement disorders like Parkinson's and dystonia.
Really?
It also has applications for Alzheimer's too.
Even better. And here we have our pathology portion of our laboratory, where we use antibodies to stain the tissue samples we've collected. In fact, Nicole's doing that just now. Nicole, why don't you tell us a little more about that?
Well, right now I'm working with one of our research scientists, Maria, and she's currently slicing tissue samples and mounting them onto slides to be immunostained. And this is in order to look for proteins of interest in Alzheimer's and Parkinson's disease.
So why don't you show us a sample of the stain?
Sure, so here is a sample of a stained tissue. As you can see, it's an indigo color. And then over here we have the unstained samples.
Really interesting. Thanks a lot. And here's the final stop on our tour, the core equipment laboratory. And here's where we have various antibodies and other techniques that we use for protein imaging and staining. Here we have one of our interns, Natalie. Natalie, what are you running today?
I'm analyzing the results obtained from a Western blot.
And what's a Western blot?
It's a technique that enables us to identify and quantify levels of proteins from different sources such as tissue or cell preparation.
That's interesting. And then how do you use it?
Protein extracts are separated by molecular weight, which is their size, and they're transferred to a membrane, then immunodetected with proper antibodies, and the signals revealed by immunochemofluorescence.
That's really interesting. And what's all that behind you?
Well, the proteins are revealed with chemoluminescence. That means they will emit light which we record with this highly sensitive digital camera. And we obtain this kind of picture representing protein in different sizes. From top to bottom, you can see the protein in different sizes.
That's interesting. Thanks a lot, Natalie. There you have it, folks. This is where young scientists do meaningful research in order to investigate the diseases that are plaguing our society. We need your support in order to make our program grow and offer more opportunities to students to do cutting-edge research at our facility. So let's go check with the running team and see what they have to say about it.
Support science.
Support science.
Support science.